Okay, so we're going back to angiotensin again. Okay, so we need to talk about, I don't know if we need to go through all this. So if blood pressure goes down, the kidneys detect it, okay, and they release renin. Okay, renin converts angiotensinogen, which is coming from the liver, to angiotensin 1. ACE comes in, it converts angiotensin 1 to angiotensin 2. Okay, now, what angiotensin 2 does, okay, it's threefold. And before we only talked about one thing. Okay, so one thing we talked about was vasoconstriction. Okay, so if it constricts the blood vessels, it's going to make your blood pressure go up. Another thing that it does is it stimulates the hypothalamus and it makes you thirsty. So if you drink, okay, that's going to increase your blood volume. Okay, and that's going to increase your blood pressure. Okay, another thing that it's going to do is it's going to constrict the efferent arteriole. Okay, and it's going to actually increase salt retention and water retention. Okay, so it's going to decrease urine output. Another thing that it triggers is it triggers the adrenal cortex to release aldosterone, okay, which we have to talk about aldosterone. I have it written out here. Okay, so angiotensin 2 comes in. It actually constricts the efferent arterioles. That increases the blood pressure in the glomerulus. It reduces the blood pressure in the capillaries. Okay, so it reduces tubular reabsorption. So we make less urine. So that's going to, if you make less urine, that increases your blood volume, and that's going to increase your blood pressure, which is what we want to do. Okay, now we need to talk about the next steps. Okay, so we've talked about filtration. Okay, so we can check that off. We're okay with that. Now we're going to talk about tubular reabsorption and secretion. And you can go over this with like really fine details, but what we're going to do is we're going to try to simplify it and make it as straightforward as possible. Okay, so the proximal convoluted tubule is designed to do lots of reabsorption. Okay, so it's all about reabsorption. Okay, so it's cool because it has a lot of active transport. It actually uses 6% of all the ATP in your body is used in your proximal convoluted tubules for active transport. Okay, so they have they're very long. Okay, it's the PCT is long. It's very convoluted, right? So it's all twisted up on itself. It has lots of microvilli, right? So remember that that's going to increase the surface area. Okay, so increases surface area for absorption, and then it's got lots of mitochondria, right, to make ATP for active transport. Okay, now 65% of the glomerular filtrate is going to be reabsorbed, okay, or picked back up in the peritubular capillaries at the PCT. Okay, so on this picture, this is representing the PCT. Well, there's the glomerulus, right? And then here's the PCT. Here are the peritubular capillaries that are going to be wrapped around it. So we're only focusing on this part of the nephron right now. Okay, so 65% of everything gets reabsorbed here. So it's going back into the blood. Okay, so we're going this way. Okay, so this picture just shows different ways that things are going through. Okay, so paracellular means it's going between the cells. Transcellular means it's coming in well, this is going the other way, but it could be going through a membrane into the cell and then through another membrane and into the blood. Okay, so we will have some secretion, like where we're going this way too. So we will have some secretion occurring as well. Okay, so, but this is the big place where everything is reabsorbed. So everything that we want to get back into the blood that we don't want to end up in the urine gets reabsorbed here. Okay, so if you remember in the filtrate, sometimes we end up with glucose. Okay, we end up with amino acids. We end up with ions. Sometimes we end up with proteins. Okay, these are things that they're like gold to us. It would be like flushing money down the toilet. Okay, so we have to get them back into the bloodstream. Okay, so they're going to be reabsorbed in the PCT. Okay, so glucose, amino acids, ions, protein. The big one, another big one would be water. Okay, so they're going to go back to the blood. We want it to go back into the capillaries. If the solutes move, the water's going to follow. Okay, so salt's going to go, water's going to follow. Okay, now, there is this thing, it's called the transport maximum and the renal threshold. Okay, so the transport maximum is the maximum amount of substance that can be reabsorbed into the blood. Okay, so if this is the filtrate, this is the blood, we only have so many channels. Whoops, I guess I should draw the channels down here. So many channels in order to get things back into the blood. Okay, if we have more than that, we go over what we call the renal threshold. Okay, so we go over that renal threshold. What will happen is the renal threshold is the maximum concentration of a substance that can be carried in blood without spilling into the urine. Okay, so the big example of this is blood glucose. Okay, so the renal threshold for glucose is 220 milligrams per deciliter of blood. Okay, so normally we do have glucose overflow. Okay, but we have enough protein carriers, okay, or channels to get it back in. Okay, so we have enough that we can get it back into the blood. But if someone's diabetic, okay, and they have excess glucose, they don't have enough carriers to get it back in here. Okay, so that it can't go back. It won't go back. So what happens is the glucose ends up in their urine. And that's one indicator that you could have diabetes mellitus. Okay, so I always think of like the renal threshold and the transport maximum. If you've ever seen I Love Lucy, and the one where she and Ethel are working in the candy factory, and they're supposed to be boxing up the chocolates, and the chocolates, they can't do it fast enough. So basically they've exceeded their transport maximum and their renal threshold. So they can't get the candy into the boxes fast enough, so it's overflowing. Okay, now, in the PCT, this table has, it describes all the ways that things are actually moved from the filtrate into the blood, or from the blood into the filtrate. And we've got pumps, we've got carriers, we have channels. I do not expect you to memorize all of these. I want you to know big picture stuff. Okay, so we're going to talk about these aquaporins more. Okay, so aquaporin is something that you should know. So aquaporins are water channels, and we'll talk about them here in a couple minutes. But everything else, you don't have to memorize these. Okay, now tubular secretion. Okay, so in the PCT, there's I'm making blood down here, which I shouldn't. I'm kind of messing you up. in the PCT, there are still wastes in here. Whoops, wait a minute. There's still wastes in the blood, okay? There are still wastes here. It doesn't all come out. So what we have to do is we have to get it back into the tube. Okay, so things that are going to move are urea, okay, uric acids, bile salts, ammonia, catecholamines, which those are neurotransmitters, and then drugs. Okay, so drugs like aspirin, anything you have to take, right? Anything you have to take every couple hours or whatever. Okay, now another thing that's going to happen is we're going to regulate the pH of the blood through the secretion of hydrogen ions and bicarbonate ions, and we're not there yet to talk about that. So hang on to that.